Oh, can everyone hear me okay? Okay, yeah, I'm starting to lose my voice. That's why I was going to use the mic here. So I want to thank the Pint of Science organizers for organizing this wonderful event combining my two loves, beer and science, really. Two things which I think go well together. Stefan Lang, who's sitting in the back here, knows that many of the experimental, and my wife also will know that many experimental ideas I come up with slightly intoxicated. So the title of my talk today was going to be Getting to the Heart of the Problem, Making the LINC Complex Less Complex. But as I was writing this a couple of nights ago, I thought, really, the title should be Everything I Ever Needed to Know About the Cardiomyocyte I Learned from IKEA. Now, that'll become more apparent as I go into the talk here, but as everyone knows, I'm a postdoc at UCSD in the Chen lab. Now, what I'll be talking to you about today is a little bit about the cell biology. Very, very little. We got a general audience here. Some people are into it, some people aren't. I'll try to keep it so we can all get on the same page. I'll talk about how the factors we look at, the LINC complex, how that affects the heart, what's known about that in the heart. Then finally talk about some of the mouse models that we work with and how they affect the cell biology of the cardiomyocyte. Now, before we go any further, why is the heart important? Or like my old graduate advisor called it the so what slide. So why is the heart important? Aside from the obvious, where pretty much you can be born without a brain but not without a heart, Dick Cheney notwithstanding. If we look at adult disease, what we can find is that cardiovascular disease is the #1 cause of morbidity and mortality in the US. Via stroke, via heart failure, via heart attack. It's incredibly prevalent. As of 2000, we spent about $972 million worldwide on treatment, on conditional medication, all that good stuff. By 2025, we're expected to spend about $1.5 billion worldwide on heart disease. This is only in the US. We spend about $60 billion annually on this. Now, what bothers me about heart disease, and the only real serious thing that I'll talk about in the slide, is that people believe this is a disease of choice. But if we look at developmental diseases, and even adult diseases, a lot of these are driven by genetic mutations that occur and cause disease either at birth, as seen with developmental diseases. Approximately 1% of all live births present with some type of developmental disease. For example, Emery-Dreifuss muscular dystrophy, which presents with cardiac defects and skeletal muscle defects, or you have patients who present later on in life with some type of mutation that causes an adult pathology down the road, including valve disease. Another example of developmental disease includes hypoplastic left heart syndrome. So all of this taken together tells us it's important both from an epidemiological and economic standpoint to understand. So our group pretty much studies the heart at multiple levels of organization. Now, my old graduate advisor, and I think several others, used to put this slide up here to kind of educate us on the levels that you're looking at. A straight reductionist view looks at a single level, but if you're looking really to try to understand what disease is and what pathology and what physiology is, you have to kind of look at all these levels right here. So to start off, you've got the molecule, which is DNA to RNA to protein. That in turn can affect the organelles, which are things like the nucleus, things like the mitochondria, things within a cell. Then you've got the cell, in this case the cardiomyocyte, which is the individual unit that performs XYZ. You've got the organ, in this case our organ of interest, the heart, if you study the kidney, the lungs, the liver, what have you, but all this goes to that organ level. And then you've got the organism level, which is in this case human, which is our main interest to kind of figure out what this is working with, or mouse, which are the mouse models we use. So essentially what we're doing is we're looking at mutations at the gene level which affect proteins, which in turn affect cells, organs, organelles, and our main focus in the lab is structural proteins within the cell. Now, most of us, when we took cell biology in high school, or actually when we looked at cells, by the way, just how many scientists do we have in the room? Okay, we're good. Okay, I can skip ahead this. I was told general audience. Okay, so we got this cell right here. Now, if we're thinking about the cell, really what we need to look at is something like this. This is a scanning electron micrograph of a cardiac fibroblast. So essentially what happened here in this experiment, this is done by Dr. Tom Borg back in 1984. What he did was basically remove the cell membrane, all of the internal cell components, and essentially all you're left with here is the nucleus, and the actin cytoskeleton here. So you've got the structural portion of the heart, or structural portion of the cell, excuse me, what's called the focal adhesion here. If anyone wants to know more in detail what that is, I can explain later. And you've got the nucleus here. Now what you can appreciate from this is that you don't just have a gelatinous mass. Essentially what you have is the nucleus, which is the largest organelle in the cell, and this strut work here that kind of builds around it, that links to the substrate, or to where the cell is adhering to the whatever it's really adhering to, be a dish, be the ECM, extracellular matrix, and then you've got this kind of strut work here that binds to the nucleus. And that's important. That's important for function, it's important for gene expression, it's important for biomechanical loading effects. So for those of you who don't study the heart, just how many people study the heart in the room? Okay, thanks, Stefan. So for those of you who don't study the heart, the cardiomyocytes are the primary contractile cells of the heart. Here you can see a confocal micrograph showing the two nuclei, which is actually unique in most organisms, that the cardiomyocytes are binucleated. Here you see the actin cytoskeleton here, and here you see a merge image, and here you see just a kind of cartoon diagram. And what you can see here, and what I drew in this diagram, was that you have unique complexes here at the nuclei which bind to the sarcomeres, which are the filaments that contract, as most of us know from our cell bio class, that actually cause the contraction to allow blood to be ejected into systemic circulation, that link this nuclei to this actin cytoskeleton here, or to the sarcomere. Now, I promised you I'd talk about IKEA. And this is kind of what I've always thought about the cell, is that this is actually, I think this is the bed that I owned prior to coming to grad school, but my wife and I bought another bed from IKEA. And what you can kind of tell is this. You've got individual components that form complexes within the overall structure. And really, if you're a dipstick like me who tends to forget putting these individual components of the complex in together, you compromise the overall biomechanical integrity of the unit. So, or if you mutate something, like if you strip, say, screw one number 00113 here, essentially what happened is you compromise, again, the overall biomechanical integrity of the unit. About 6 months ago, my wife and I tried to put one of these together, and many swearings later, I realized I had forgotten a single component of the complex, which again is a mutation deletion, or can be seen as a mutation deletion. When I was in undergrad and I had one of these beds, I forgot a screw again. And what happened when I did that? I jumped on the bed and the entire thing fell apart. So if we think about the nucleus, if we think about how the nucleus actually binds to the cytoskeleton, what you can see here is kind of a blown up cartoon diagram of the nuclear membrane. This is a double membrane here, seen here the outer nuclear membrane and internuclear membrane. And you see a complex here termed the LINC complex, which is the linker of nucleoskeleton to cytoskeleton complex, which physically links that strut work that I showed you earlier, the actin cytoskeleton, to the internal nucleoskeleton here, the lamins, which actually reside right underneath that internuclear membrane and actually give that structural integrity to the nucleus. So here what you see are the nesprins, primarily nesprin 1, nesprin 2, which are giant, about 1 megadalton. If you ever run a Western, you'll know what that means. 1 megadalton or 967 kilodalton proteins that reside at the outer nuclear membrane. They in turn bind to sun proteins, and that in turn binds to the lamins. What this by itself tells you is that, okay, this is important. This has to be important. Something has to be going on here. And indeed, if we look at the hypothesis of force transmission, we can see that actually something is going on here. So imagine this cartoon diagram here being our bed. I jump on bed, biomechanical loading, something happens in the nucleus. If I forget any of the components here, mutate or delete or somehow change any of the components along this continuum, in this case being the extracellular matrix, or the matrix outside of the cell membrane, the actin cytoskeleton, or these nesprins that reside at the outer nuclear membrane, if I change anything here, you could have disease. And essentially what the hypothesis was early on in this field was that these were playing a major role in the overall nucleus. And so about 5-10 years ago, people started looking clinically at Emery-Dreifuss muscular dystrophy patients, and out of 160 patients, they found about 10 of them that had mutations in SIGN 1 or SIGN 2. These are the genes that encode for the nesprins. What we also found a couple of years back was that, not we, another lab in Chicago found, was that There was a patient who had cardiomyopathy, or thinning of the cardiac walls, reduction in pump function, that showed that they had mutations inside of this Nesperin. So again, their screw was stripped or the screw was gone. Some way, these Nesperins were causing some type of cardiac defects, but we still didn't know what was going on there. We still didn't know if it was a force transmission thing, a biomechanical thing, a biochemical thing. We had no idea. So fast forward, and again, I'm simplifying a lot of this. There's a lot of literature there, and if anybody wants to ask me, we can buy me a beer later, and feel free to talk to me about this. But the study aim that I've been kind of working on for the past 3-4 years is to understand the roles of Nesperin 1 and Nesperin 2 in the heart, in the cardiac muscle cell. And the way we designed this, and it just recently got published, the way we designed it was making mouse models. You know, we work with mouse models in our lab, that's our model. So we had a mouse model where you have both Nesperin 1 and Nesperin 2, a mouse model where only in the cardiac muscle cells you lose Nesperin 1, a mouse model of Nesperin 2 loss, and a mouse model where you lose Nesperin 1 and Nesperin 2 in the cardiac muscle cell. Okay, so to recap, you've got a cardiac muscle cell. You've got these links, 2 arms, binding the nucleus to the actin cytoskeleton. And so what we've done is delete one arm or the other, delete one of these factors. And again, we wanted a phenotype, so check for cardiac defects. We did a lot of measurements. It's in the paper, but what I'm going to show you today is echocardiography. So if anyone's actually seen or had an echo done on them, if you've ever had a child, or if you've ever had an echo on your heart, we essentially did that with the mice. And then I'm going to show you some nuclear shape and positioning studies, and then show you a biomechanical loading study that we did with the engineers at UCSD. So what we did was perform first mouse echocardiography. Here's that base setup. You have the mouse. Again, the poor mouse is on the table. I think it's a rat, actually. You have a probe, and that measures the echocardiographic function of the heart. And what you can see in this, I'm really simplifying here so I don't run out of time, is that over the life of the animal, from 5 weeks to 10 weeks of age, that when you have both Nesperin 1 and Nesperin 2 deleted, you actually drop pump function. So by deleting both of these factors in the heart, you actually get a weaker cardiac pump. Now, that's kind of at the organism level. Let's look at the cell level. So this is, the slide's kind of busy, but basically what we did was isolate our cardiac muscle cells, seen here in these green images and blue, and look at the nuclei seen in the blue. Now, what you'll see is that in the normal cells, you get these nice, happy, round nuclei that are dispersed evenly throughout the cell. When you delete Nesperin 1 or Nesperin 2, so lose one of the arms, they become elongated, elliptical, and are kind of varying sparse throughout the cell, both in Nesperin and Nesperin 2. When you delete them both, what winds up happening is they become elongated and really close together. So we want to look a little bit more in detail, because this was kind of a weird result. And when we looked at some electron microscopy, any electron microscopists in the room? Okay, cool. I'll be bugging you guys later to get more info on this. I'm not an EM person by any stretch of the means. But when we looked at this, what we saw was that the normal nuclei was kind of round, kind of had this like dark patch around the circle, so nice, happy, normal nuclei. The single knockouts were kind of invaginated, or they were elongated, you know, nice, a little bit different, but not super different. And the double knockout kind of looked more like this wavy guy right here with these dark patches coming out here. So that told us there was a cell phenotype there. That when you lose the 2 links to the cytoskeleton, to the sarcomere, both there's a organism change and now a cell biology change. So we want to see if there's any type of mechanical loading change. And we did this in collaboration with Andrew McCulloch's lab at UCSD using homemade biomechanical stretchers seen here on the far left. So you've got these stretchers here. You've got this kind of groove pattern substrate here, or this groove pattern plating area here. You put your isolate your cells, you put them down, they become nice, happy, rod-like, and then you stretch them 14%. So essentially you're adding that load. So remember when I jumped on my bed and it fell apart? Essentially we're jumping on the bed here. We're trying to jump on the bed and see exactly how these cells respond. Now when we did this, what was kind of cool was that we looked at gene function. Now there's marker genes every time something, in some cases, that can really provide you a good bit of information. So say, for example, I stretch a cell. We're going to think that EGR1 and c-Jun, which are just 2 individual genes that have been shown to be induced when you jump on the bed or stretch the cells, go up. And they did in both both condition or in the wild types. When you deleted Nesprin-1 or Nesprin-2, you got a reduction in response. But when you deleted both, response gone. So we kind of through that, and I'm running short on time here. We built this model here, and this is in the paper. And if anyone wants to know, again, buy me a beer and I'll talk to you more about it. In that, in the cell, you've got the sarcomere set up here. You've got the nucleus here. You apply a load with normal nuclear morphology and positioning, and you get normal function. Happy normal. When you delete these two, Nesprin-1, Nesprin-2, what you get is an elongated nuclei. When you apply load, nothing happens, and you in turn get cardiomyopathy. And I didn't show this here as increased fibrosis. So we're trying to figure out what this means more in detail, learn what the mechanism is, because this actually does mimic some human mutations. And another arm of the lab, which I'm not involved in, we're actually going to look at specific point mutations to mimic human disease. But one of the projects that I'm kind of working on right now is a more detailed analysis. And this is thanks to Illumina. They gave me a small, like 10 grand to do this grant, or to this project. We're going to look at biomechanical loading and group the changes and look at individual chromosomes. We're going to try to identify new gene targets, do all of that good, do the good hardcore molecular biology and using next generation sequencing to figure out what's going on there. So I've talked 15 minutes, so I just want to acknowledge a couple of people. Stefan's in the back. He's right here. He's right there. He's really helped out, but I also want to acknowledge, first off, my mentor, Dr. Ju Chen. Ju's been a great mentor. He's provided a lot of opportunity for me to work in the lab, for me to play with this project. And again, I'm summarizing about 2 years worth of work in 15 minutes, so I'm going a little quick here. The people here on the left, Jalin, Matt, Jen, Karina, Kung Fu, and Andy, all really helped me out with the project. Sylvia Evans' lab and Tom Morris give me a hand. Vishal Nigam and Tom Borg really helped out. Andrew McCulloch's lab, Kyle and Emily both really helped out with the stretch studies. But I really want to point out these two right here. These are my two undergrads, Michelle and Steph, and they're both working in the lab right now. They're both phenomenal. The AHA for giving me some money to do this project, same with Illumina, and then kind of give a plug to the Alternative Muscle Conference, which Stefan and I are organizing, which is going to be later in the summer. You can look that up online, and I know it went kind of fast, but this is the real ruler of my house, Ghost. And yes, I'm a Game of Thrones fan. So if anyone has any questions, I'd be happy to answer them. I know I went kind of quick in the talk, but thank you for your time and the beer.
Questions?